Hi guys, how are you doing? I hope you're doing very nice and you enjoyed our previous videos and the lectures. And today we will talk about our next tutorial about the title of the tutorial is about how to identify a research target, a research topic to study. So when we talk about research, we talk about three things, right? We talk about DNA, we talk about RNA, we talk about proteins. On this earth, whatever you can study in biological sciences cannot go beyond anything more than this. Either it will be a DNA molecule, or it would be a RNA molecule, or it would be a protein, and I'm talking about in a very general way, okay? And how people study these things. I want to tell you about different possible ways to study the biology. Okay, let's talk about DNA. So the DNA means a chromosome. Okay. And you know the chromosome is made up of DNA and proteins. What you will study here, either you will study DNA, how the different structures, how the type of different types of DNA, how these different types of DNA are regulating the gene expression. At the same time, you will study the proteins. So there are different types of protein which are present in chromosome. Some are the structural protein like histone, some are the functional protein like transcription factors, some other protein like RNA polymerase, some other proteins like DNA polymerases. So what you will study in this area, in the area of DNA, is the DNA structure, the function of different types of DNA structure, how the DNA can regulate the regulation of gene expression, etc. And you will study about the proteins, which kind of proteins are involved in the DNA, are interacting with the DNA, the interaction, remember this term, interaction, okay? And the interaction is a kind of non-covalent interaction. Something comes and something interacts and something goes. It's like, for example, take these two objects, okay? Take this as a DNA and take this as a protein. What I mean to say is interact. For example, at some situation X and these protein and DNA need to interact, okay? Now, so the function, whatever, so they have interacted to perform particular function. Now the function is complete. And now these two molecules are separated, and now they will easily separate. So this is a kind of non-covalent interaction. They will interact whenever it's required. So in chromosome you will study about DNA, as I said, and the protein you will study about what kind of proteins are interacting. For example, the histone proteins, they help to pack the DNA in the form of compact packing in the form of chromosome, right? And there are some functional proteins like transcription factor, induce the transcription of a particular gene when it's required. RNA polymerase, you know that polymerase synthesize the mRNA. DNA polymerase, which will replicate the DNA, and etc. So in the field of DNA, you can only study about these things. Then we'll talk about RNA. What you will study in RNA? You know about the ribozymes. So the catalyzing RNA, so in the field of RNA, you will study about the types of RNA which perform the catalysis, which perform the enzymatic reactions. You will study about the RNA protein interaction, how the RNA and protein interact to facilitate some functional role. I mean, either they will regulate some gene expression, or the RNA, the mRNA interact with protein and form some kind of RNA protein complexes, which will then transport from the nucleus, where the transcription happens, to the cytosol, where now this mRNA will be used to translate protein. And another could be the splicing machines. So you know about the third function about the RNA is a splicing machine. So there are different type of regulatory RNA perform their role in the splicing, RNA splicing. So If you don't know about the RNA splicing, you can just simply search in the Wikipedia about the RNA splicing. It's a very important process. So these are the things which you will study in RNA, and the third and the fourth category is like types and function of different RNA. So like microRNA, like so it's like microRNA and a small nuclear RNA, and several many types you will definitely find in one of our article. So these are the things which you can study in the field of RNA. In the field of RNA, one more thing which is possible is DNA RNA interaction. So you know that during the process of transcription, when RNA polymerase use the DNA as a template and synthesize the messenger RNAs and tRNAs, rRNAs, etc. So how this DNA RNA is interacting there. So this is another branch which you can study in the field of RNA, and the whole branch of the protein. For the protein, there can be the structural protein, functional proteins. So the proteins, I mean, there are different ways to categorize a protein, but I will definitely generally characterize here from the two bases: the structural protein, which form the cellular structure, which maintain the structural component inside the cell, and the functional protein are the protein which perform some enzymatic functions, interactions, cell signaling, and etc. So around 90% of research around the world is focused on studying by any means the proteins. So most of like around 90% of research is focused around proteins. They want to study how these proteins function, how these proteins interact, how they regulate, how they catalyze a particular reaction, etc. And we know by our textbook that proteins are the structural, are the building block of the cell. And that's how they are building block, because they not only perform the structural function, but they will also regulate the function, regulation, etc. And then the protein which we can study is by their, of course, their structural role, their functional role, etc. Cell signaling, and this is one thing. And on the other hand, you can study the, as I said already, the RNA protein interaction, DNA protein interaction, and protein interaction. So these are just kind of very broad way to introduce you about the different domain of the research and what are the general possible projects or the area where you can study. This is just a very general introduction, and this is all which I want to introduce you here for this tutorial. And please do let me know if you have any questions or any confusion. Feel free to ask, feel free to write me a message, and we will resolve your query within 24 hours. So don't worry, we can make it together, and just give your feedback if you want any improvement or anything. Thank you very much.